Okay, so we're gonna do a rat dissection using a really, really old rat. This has been in this nasty fluid for a really long time. Hopefully it looks good on the inside, but we will find out. So, get your dissection sheet. I'm gonna work my way along it. Go through the directions. Secure the specimen ventral side up. Now, normally you could pin it down if you want, but I'm kind of used to this, so I'm gonna do quick. Dissections work much better with scissors than they do with edges. Okay. Because there's a lot of layers to go. Once you get through the layers, though, you can put the scissors up against the inside and snip without hurting much of anything. That's the liver coming exposed there. We're going to start with the abdominal cavity. It's one thing I like with not securing it is that you can just turn it and cut from whatever angle makes sense for you. We will eventually go into the chest cavity, so while I'm at it, I'm going to do that as well. Yeah, this has definitely been in the old preservative fluid, which is nasty, nasty stuff, even though I've been soaking these in water for a week. Fairly nasty stuff. Okay, one thing I did not put in down is, and what the heck is it? Oh, you got it. Most people would be doing this with gloves on, which is fine. I'm just used to doing it without the gloves. So, working on my way to question 1. This is the peritoneal membrane. It encloses the whole abdominal cavity. There's also a membrane that holds the guts together, very fine, shiny membrane. That's the mesentery. In human beings, when you start to put in more calories than your body is using, it will take the bits and pieces and go, okay, we're going to use this energy to make fat. And if you're males, if you shoot me for a second, if you're males, the first place it tends to go is right here. This is purely for demonstration purposes. That does not tend to be the first place it goes in women. It goes here. Now keep in mind, putting fat here does nothing to the hip bones. So people go, oh yeah, well that's for childbirth. No, it doesn't make your hip bones any wider. It just makes them look wider, which is actually relates to the bonus question. But the first question is, why does the fat go here in men primarily and not here in women? And then the bonus question is, so when women do this, it's a deceptive secondary sexual characteristic. And for the bonus points, you can tell me what that means and why it works that way. Okay, come on back in. This is the liver. It's got lots and lots and lots and lots of jobs. The lab goes through the lots and lots and lots and lots of jobs. That leads into one of the dark questions on the lab. So why is the liver one of your biggest internal organs? The liver is split up into lobes. See, it's a section here, and a section here, and a section here. Let's see what we got. We've got a section, let's see, that was 3. This is
I
this.
didn't get that. Start
Okay.
it. And I'm mostly getting your fingers.
Okay. 1, 2, 3, 4, 5. It does vary from animal to animal, and when my other class does fetal pigs, there's fewer of them in a fetus than there is in an adult. But looks like we got 5, and that's the next question: is how many lobes do you get? Now, the next tricky question is, so why split the liver up into these different sections? And the first thing that always occurs to people is, well, each one does a different job. It does a bunch of jobs, must do different ones in each lobe. Keep in mind, human beings. If this one did a different job than this one, it would have a different name. If this one did a different job, it would have a different name. They do not do different jobs. They don't do the jobs in a particular order. Is that every single lobe does all of the jobs. In fact, if you need a liver transplant, you can get a single lobe and be okay. Now, so the answer isn't functional. It has to do with the structure underneath. That's the clue. Remember, this is the left side of the rat. This is the right side, even though it's on the left is on our right and right's on our left. And we will identify that structure pretty quickly if you don't know what it is right now. But what's not underneath the liver, what would be right here in most mammals, is the gallbladder. The gallbladder stores bile. When you eat anything fatty, it comes up to your body temperature and it tends to liquefy it. It becomes oily and blobs up. And because fat and oils are hydrophobic, they blob together. But we have to digest them from the outside in, because that's the way our digestion works. We can't get into it. We have to work our way in. And a big blob is all the way through your system before you've digested it all. So you don't want big blobs of oil and lipids going through your system. Bile is an emulsifier. It makes the blobs break apart. It doesn't break the molecules apart. It's not digestive. It just breaks the blobs apart so that you have little blobs that you can digest as they go through the system. Now, in human beings, sometimes you have problems. The bile salts form crystals. They block up the tubes, and you may need to have your gallbladder taken out. Now, digesting lipids is really, really important. And it turns out, without the gallbladder, you can still do it. It isn't that big a deal to have your gallbladder taken out. And your next question is, so why not? If bile is a big deal, then why does having your gallbladder missing not really mess up your digestion? And I did actually tell you as I went through that, and it's in the lab if you read very carefully. Now we're going to get in and start taking things out. We're going to get the liver out of here, snip, snip. Most of these organs are not being held in by too much. A couple of membranes, a couple of supporting ligaments, and that's pretty much it. So there goes the liver. Here's the stomach. Let's see. That's another liver lobe. There were 6 of them. Remember I said there's often 6? I missed it. It was such a flat disc. Okay, so here's the stomach attached to the outside. Well, not so attached in this guy. Is this long, flat thing. It looks very much like liver, but this is your spleen. And I can cut this out. This is a big bag of blood. This is your blood storage organ. If something happens and you're doing a decent amount of bleeding, it has red blood cell backups to dump into your blood. It's where a lot of your white blood cells are stored, so that if you run into a big infection or damage issue, there's a bunch of white blood cells that can come flooding into your blood from your spleen. It also is very thin, has a very thin lining on it, and it is very easy to break it open with an impact. Is that our spleen is over here, just under these floating ribs, not that well protected. And an impact can literally crack the casing. They call it rupturing your spleen. And as you can imagine, rupturing a blood storage organ inside your abdominal cavity, not a good thing. You can bleed to death that way. But fixing the spleen always used to be very tricky because it's very thin, and sewing it up could be like trying to repair an old curtain. It would just tear as you tried to repair it. So a lot of people would have their spleens removed rather than letting them die from the blood loss. They have a special type of surgical tape. They actually tape over the cracks now. It works pretty well. So people don't have their spleens removed as often. But say you had an accident a few years ago, and you had your spleen out, and before COVID, you were planning a trip to canoe up the Amazon. So you go into your doctor going, I'm going to the Amazon, doc, I need all of my shots. Your doctor, if he was a good doctor, would go, you're not going to the Amazon. You're not going anywhere near a third world country. And you'd go, why, doc? And he'd go, because you don't have a spleen. And you'd go, well, what does that mean? That's the question. You're gonna tell me why the doctor won't let somebody, go canoeing up the infested Amazon with, without a spleen. Okay, now we've got the stomach. I'm gonna do something that the lab doesn't necessarily specify. I'm gonna take the stomach out because it'll show it better. Take it out, take it out. Okay. This is the esophagus coming down from the throat. That's the entry tube to the stomach. This is the small intestine, the exit for the stomach. You notice when I squeeze it, it's kind of pressurized. The first thing you want to do is look at the lining, and that takes a very gentle snip, because if you squeeze it and snip, you wind up being bathed in whatever was in the stomach. Okay, I'm gonna go wash this out because there's food in here. Don't have to deal with this with fetal pigs, they haven't been eating. Okay. So this is the stomach lining. If the light's bouncing off right, you can see it's kind of bumpy and ridgy. Usually the ridges are more pronounced than this. It may have something to do with this being a really old rat in terms of having been in the preservative a long time. So your next question is just to describe the lining. Your lining is probably a lot bumpier than this. And then the next thing is to look here where the tube comes in and look to see what kind of structure that is. How clearly can you see that?
I'm not sure. Hopefully clear enough.
And here, where the tube goes out, there's another similar structure there, not as easy to see, right there. Now these are both two versions of the same kind of structure, is that you have other types of structures like that, other places in your body. You need to figure out what kind of structure you're looking at, and I don't want the technical terms, I want to know what type of structure that is. And then another one of the tricky questions, what are they doing here? And it's a slightly different answer for the one at the entrance to the one at the exit. They are both connected to the same feature of the stomach, but the one at the entrance is doing a slightly different job than the one at the exit. Okay, on to the next page. This is why I get labs from people with all sorts of gunk on them. Now we've got intestines, held together by the mesenteric membrane. You can see right there that it's kind of shiny. And you can see all the blood vessels in the membrane. Is that a lot of food is being digested in here. The nutrients are going into the blood or into the lymph drainage tubes and being all carried eventually into the liver. The liver is a big detox part of your system. So stuff that comes in from your digestive system that isn't really ready for distribution, it has issues, can be dealt with by your liver. Okay, the next thing is we're gonna unravel this. Now this particular, one nice thing about this being a really old rat is this membrane is really more fragile than it typically is. So I don't really need to snip it so much as I can tear it. Now right here, this blobby area right here, and it's part of this also, is part of the pancreas. The pancreas very rarely looks like the nice leaf-shaped structure that you see in pictures of what a pancreas looks like. It's very often much more kind of spread out all over the place and kind of hanging in the membrane. I think that's where the Whipple procedure comes from, where if you have pancreatic cancer, you can't just take your pancreas out because it's doing too many important things. But I would assume in people, I don't know this for a fact, I would assume in people a lot of times it's not all one pancreas in one place, and you may be able to take part of it out and leave part of it behind. But I've never actually looked up to see what makes the Whipple procedure work. And if you're interested in going to med school, you may learn that eventually. I picked up the term from Gray's Anatomy. Well, I'm starting to think I should age these rats. This is much easier to do than it typically is. Okay, what I've got a hold of right here is a dead end sac called cecum that is very different sizes in very different mammals. This is much bigger proportionately than we would have. There's more of the pancreas. You can see that blobby white stuff in the membrane. And this, now it doesn't want to tear. The cecum is a fermentation sac. So in animals that eat a lot of plants, it can be huge. If we were doing a rabbit, this would be a foot long. But in a rat, it's a decent length, but it's not huge. Okay, let me flip this over. Just give me a second here. Okay, so here is technically where I've got it pinched between my thumb and my finger. This is actually the beginning of the large intestine, the colon. But right where I've got it pinched with my other thumb, that's the end of the small intestine. And your next question is, so how do you know you've reached the end of the small intestine? And the answer should be in the way that this fits together. It's like I always tell my students when they're looking for it, imagine you're on a road, and when you hit the end of the road, there'd be no question you hit the end of the road. And there's the end of your road. So I'm going to separate this out. This is the small intestine. And the next thing we're going to answer is, there's still a little bit in here. Not entirely sure. There's more pancreas. Ah, there we go. Okay, I'm going to be your lab partner. You don't need to follow me. I'm going to lay this out. It is 87 centimeters long. For an animal that is about 18 centimeters long. So decent length. This is the large intestine. Now, rat fecal pellets are very kind of big and oval. If you've had a rat, you know that. And you can actually see them forming in the colon here. Oops. Snip. What have we got in here? There's the very end. Snip. This, of course, is much shorter. Including the cecum, about 17. So actually close to the body length. There's no rule like that, but this turned out to be. And surprise, there's another liver lobe. Is that, as I said, it varies from animal to animal. They wind up being tucked all over the place. That's kind of a clue as to why they're split up into lobes. Okay, the body cavity right now has got too much fluid in it. I have got to flip my page here. We've got kidneys on each side. Now, this rat did not read the books because this kidney is supposed to be lower than that kidney because the liver tends to push it lower as they form. But this kidney is actually more anterior, that's toward the head. and this one's more posterior. And that's the thing, that's one of the reasons why I'm a big believer in dissecting actual animals as opposed to models. Because you get to see the variation, the things that any animal you grab is gonna have something about it that isn't quite the way the book wants it to be. Okay, what have we got here? So we're gonna take a kidney out. And it really does look literally like a kidney bean. In the rat, it's virtually the same size as a kidney bean. In us, of course, it would be much larger. And now, one of those things, those tricky things to do, which is we want to cut it in half without cutting us. Getting a nice, right through the middle cut. Okay, let's set this on the paper here. There are 3 layers to this. There's an outer layer that's just the epithelium, it's just the capsule. So if you draw the outline of this kidney, the line you drew it with is the capsule. This layer underneath, and I don't know if the light's reflecting off of it, the way I'm seeing it, there's all these lines running out toward the edge. That is the cortex, where the filters and the drainage tubes are. In here, that's kind of redder and blobbier, is the medulla, where a whole bunch of stuff is collected. And this shiny part here, it's visible more on this side than on that side, is the central cavity, where everything's collected and it goes out through the ureters to the bladder. A lot of times this is so flat, it's easy to miss it. I'm kind of surprised that I caught it with a kidney this small. How far in are we?
22 minutes.
Okay, we're gonna stop there on this video. Pick up the next one.